safeguard product quality across the pharmaceutical supply chain from principles to practical real-world application. Okay, it's my privilege to introduce our speaker today, Mr. 𝐓𝐢𝐤𝐸, Senior Manager International Regulatory Policy and Intelligence at the US Pharmacopoeia. In this role, he works as the intersection of global regulatory policy and pharmaceutical quality standards, engaging with regulatory bodies and industry stakeholders to strengthen supply chain integrity worldwide. His perspective brings together international regulatory intelligence with practical on-the-ground insight into how storage and distribution practices are evolving to meet today's compliance demand. We are glad to have him share the expertise with us today. Okay, I'm sharing the screen now, and for your info, it's a recorded webinar. So if you have any queries, you can email us. We are going to share to you the email address.
Today, I'm going to talk about good storage. Thank you so much for joining me today. I'm 𝐓𝐢𝐤𝐸. I'm heading the International Regulatory Policy and Intelligence Unit at United States Pharmacopoeia. So today, I'm going to talk about good storage and distribution. Specifically, the GMP, it connects the GMP with the real world, where the medicines reaches the patient. And I always says that this is the area or this is the avenue where the quality meets with the reality. Let me share my screen for the presentation. Okay, so before I define GSTP, I will ask you one question. What happens to a medicine after it leaves the factory? Think about it. Clinical trials, millions in the manufacturing, robust GMP audits. But when the medicines is packed and shipped, it enters into world of drugs, airports, custom warehouses, and last mile courier. I will share few interesting but shocking facts in this slide. So single temperature excursion during the distribution of a CAR T therapy, single CAR T therapy. We are not talking about multiple. Single CAR T therapy can destroy the shipment of which is costing over 500,000 US dollars. Single shipment. It is also estimated that half of the global vaccine are at the risk of wastage due to the cold chain failure. In 2024, alone UNICEF distributed 2.8 billion worth of, not worth, I'm sorry, 2.8 billion doses of the vaccines in 99 countries globally. So GSTP is the quality system that protects the medicine in this space. So for the ease of understanding, I have divided my presentation into a simple framework, the SHIELD framework. So 6 words, 6 sections. So S stand for the standards. So in this section, we will study about what are the GSP standards, where it comes from. H stand for the high-tech tool, in which we will study about the advanced tools and techniques to make the distribution good. I stand for the immunization. We will have a deep dive about the vaccine supply chain. E stand for engineered product, cell and gene therapy. In this section, we will see what are the some delicacies for this kind of a product supply chain. Then the L stand for linking these distribution data with the patient safety. We will see how we can do it. And last but not the least, these stand for deliver the change. How you can implement practically in your daily routine if you are a pharmaceutical professional working in different fields. So let's begin with the standard. Yes, this is the basic. So GSTP stand for the good storage and distribution practices. This is a WHO term. In European Union and PICS, which is the Pharmaceutical Inspection or Cooperation Scheme, we have different terminology which we call GDP, good distribution practices. Although the names are different, but the concept and idea is the same. So GSTP ensures that the quality, safety, and efficacy of the medicines are maintained for the manufacturers till all the way to the patient. So there are 4 important regulatory reference you must know. Number 1 is the WHO TRS 1025 and X7. This is the WHO guidelines for good storage and distribution practices. This guideline focuses on the risk management and quality system throughout the supply chain, including the community pharmacies. The concept in this guidance document is applicable for both developing countries and developed countries. Next comes to the European Union or EU GDP guidelines. This is a legal requirement in European Union member states. So this guidelines introduce the concept of a responsible person, like qualified person in GMP. This guidelines introduce the concept of responsible person who holds the responsibility for the GSTP or GDP compliance. This guidelines also focus on the qualification of the third-party logistic provider and brokers. Then comes to the PICS guidelines. So this guidelines is inspection-focused guidelines. So it primarily talk about what are the areas that the inspector will be looking during the GDP inspection. So it's important to learn and prepare the facility accordingly. Then comes to the USP Journal Chapters 1079. This is a US standard, and it focuses on the appropriateness condition of during the storage and distribution. So GSTP has a quality system and must have a quality system. It primarily comprise of the 6 key pillars. So I will not go into the details. On this slide, I will just discuss 2 important things. So in the GSTP quality system, all these systems are like you might be familiar with the GMP um, quality system. So similar pillars in the GSTP quality system. However, there are two important points that I want to emphasize on this slide. One is in the GSTP operation, apply the risk-based approach following the ICH-Q9 and Q10 principles, which include the risk metrics and FEMA methodology. So another important point that for your quality system, you must have an internal audit, not just for your compliance against the established standard, but also um, you must audit the external service provider, specifically the third party logistic providers. This is a missing part in most of the GSTP quality system. And it is very well reflected here that you can outsource the work. Yes, you can outsource the work by hiring a third party service provider, but you cannot outsource the responsibility. So that's why you need to make sure that all of your service provider should be audited and qualified before giving any contract. So this is a typical supply journey of a medicines. It has the manufacturers, primary warehouse, regional distribution centers, last mile delivery, courier services, and then finally the patient. So five primarily the stages. But in this slide, I want you to focus not on the stages itself, but the handoff between these stages. From manufacturers to primary warehouse, there is a handoff, and so on, so on. So whenever in the supply chain, when the product switches hand, so there is a risk. And risk could be like during the handoff, there could be temperature excursions during the loading, unloading. There could be physical damage due to poor handling. And most importantly, data discontinuity. Whenever the product being handover from one partner to another partner, there might be scenarios where the data about the storage conditions are not carried over to the next partner. And last but not the least, the accountability gap. When there are multiple stakeholders or multiple players involved in the supply chain, so there could be a moment where nobody is sure who is responsible. The manufacturer is responsible, or the middleman, third party is responsible, or who is the responsible. So whenever the product switches hand within the supply chain, there is a risk. Why we are talking this? Because when we are designing risk-based approaches, we must pay attention to these hands-off. So GMP is the quality standards for manufacturing, and GMP controls the quality before the product is released to the market. And GSTB is the natural extension. GSTB control everything once the product is released or transported in the market. So, but however, there is a very misconception about GSTP. Most of the manufacturers or stakeholder believes that their product is very much stable and they should not be worried about the distribution challenges or distributions discussion about the temperatures and rest of the things. But in fact, the reality is that stability is consumed. Stability is consumed cumulatively. Every discussion across every partner use a portion of a stability budget. It's a very interesting phenomena we will see in the upcoming slide. Okay, now we comes to the second part of our presentation, which is H high-tech tools. So if you want to remember one concept from today, please make it this one. This is a very unique concept. I love about this concept. It's hard to implement. And let me try to, explain in a simple words. So we know that every medicine has a printed expiry date. But there is another thing which I call a shadow shelf life. This is the real remaining stability after all the temperature excursion on the airport wait time or custom warehouse delays. And you might be thinking how this works. So imagine a bar. As it could be a battery indicator on your phone, or let me try to, yeah. So that bar, imagine that bar represents the total cumulative time the product can spend outside the specified storage condition. I'm emphasizing this. This is very important. So this total bar represents the cumulative time that a product can spend outside the specified storage condition. Whenever, during the supply chain or distribution, the as recalling from the previous slide, there is a handoff, the medicine changes the hands. So a portion of this stability battery is consumed. Like as you recall, during the loading and offloading, we cannot ensure that the specified storage condition are met. When in transit, yes, there is the transporter, even on their best, they want to ensure that the storage condition are maintained, but still there is uh, a small portion where we cannot ensure the specified storage condition compliance. So every time the product is exposed to the storage condition or the specified condition which is beyond what is written or on which it is not stable, so it consumes a stability. It's like a resilience. The product can withstand um, against the external temperature excursions. It's not just the temperature. Sometimes it's vibration. Sometimes it's freezing. We will see in the next slide. So this and the uh, challenging part that not a single entity has a full picture that how much stability budget has been already utilized and how much is the remaining. Specifically for the biological product, uh, the numbers are very tight. 60% of the biological have like room temperature stability of just 1 week to 1 month. And you might be wondering, okay, how we can, this sound good, but how we can calculate? So we have the practical tool, which we call TOR or TOS, temperature, the time outside the refrigeration or time outside the specified storage condition. Okay, the next important tool is lane risk profiling and lane qualification. So lane is the shipping route from point A to point B with all the specified mode, carrier, and conditions. So lane risk profiling is the assessment of the risk associated with each shipping route or lane. So the process for the lane risk profiling is very simple. We have to first assess the product risk itself. How much sensitive is our product? It could be like very sensitive to the environmental condition. It could be like stable. Then comes to the lane risk, which include whether our shipping route crosses different climatic zone or it's just one climatic zone across whole the supply chain. How much is the transit time? How many are the hands off? These all include in the lane risk. Then comes the process risk. Have we a written procedure for every operation within the distribution, or it's not a harmonized approach? whether the staff is trained on those SOPs, and very importantly, the partner capabilities, because your organization might comply with the GSTP quality system, but your partner, the third service provider, they may not be having the same capabilities or same quality system. And last but not the least, the equipment qualification, the monitoring devices equipment qualification. For example, the hygrometer, the thermometers, and so on. Once you have the lane risk profiling, then comes to the lane qualification, which is running a test shipment across your lane. It's of course uh, attached with the temperatures monitoring across, throughout the supply chain, and we have to do the lane qualification across different seasons. In Brazil, the Anvisa, which is the national regulatory authority, requires 3 seasons lane qualification for the biologicals. This is a global best practice. And whenever you change your lane, your shipping route, your carrier, your mode, there must be a change control because you already qualified your lane. Once you make any change, you need to qualify again under the change control. So over the past many years, the monitoring landscape has evolved dramatically. I must acknowledge this. So the supply chain monitoring used to be a reactive approach. In reactive approach, we put in the data logger in the boxes at the final destination. We opened the box and saw the bad news. So this we call the forensic logistic. But nowadays, we have IoT devices, which is Internet of Things, which transmit the real-time data about the temperature and the other parameters. And based on that, we can reroute, we can expedite the shipment, even we can quarantine the shipment before the product is lost. So we call proactive, and we are switching from proactive or predictive kind of a analysis. So this is a key shift from discovering the damage approach to preventing the damage approach in the supply chain. So in the bottom, you can see there are some important references. WHO PQS E006. This is a WHO standard for pre-qualification of the temperature devices, cold chain devices for to be used in the immunization. And one important another reference on the second, the control and go. This is a IoT pre-configured IoT devices validated for GMP, and over 200 airlines already approved this now. So this is IoT devices for the proactive or predictive kind of a monitoring during the supply chain. Here comes another interesting tool, supply chain control towers. These are the centralized digital command centers providing end-to-end visibility across the pharmaceutical supply chain. Think air traffic control, but for the pharmaceutical. It has 4 capabilities. Like the supply chain control tower can track shipment across multiple carriers and geographies in the real time. Based on the machine learning model, it can predict the delays, temperature excursions before they occur. And based on the anomalies, it can automatically reroute the decisions and escalation protocols. Last but not the least, it has a data um, integration is possible across many platforms. However, still we have data standardization challenges across different partners, and cost of the implementation remains a barrier for the smaller organization. However, the high value products like the cell and gene therapy, the investment pays off. Now comes to the serialization and track and trace. Serialization is identification or giving a unique identification number on each pack. Both US and EU require serialization, and nowadays more than 70 countries has a serialization and track and trace requirement. So serialization actually connects to GSTP, and it helps in the targeted recalls, distribution traceability, and counterfeit detection. So serialization is the infrastructure behind the safety surveillance. Now comes to another session, which is immunization. So in this session, we will see vaccine supply chain and where GSTP is the method of uh, life and death. So let me share a scale, although we have discussed in the beginning of this session, that in 2024 alone, UNICEF distributed 2.8 billion doses across 99 countries. And distribution system spans from the national cold chain warehouses to the community level health workers for the day-to-day routine or day-to-day immunization campaigns. And the transportation mode included like refrigerated trucks, motorcycles, even donkeys, drones, and walking. So let me quickly share a very famous case study, which we call SMOA case study 2019. So what happened in SMOA in 2019? Two nurses improperly constituted a MMR vaccine. Instead of using a diluent water for injection, they have used muscle relaxant. So diluent choice was wrong, and also that diluent was expired. So what happened? Two infant died immediately. In response to this, the national immunization program suspended. What happened due to this suspension of the national program? The coverage collapsed from 58 to 31%. And due to this consequence, 83 additional deaths from the measles caused by the suspension, not from the original incidence, but from the suspension. So the key lesson here, a single distribution quality failure in the vaccine can trigger a public health cascades that kills more than the original incidence. So GSTP is not just about refrigerators. It's a chain of trust from the manufacturers to the patient. And if the link is broken, there is a cascading effect. So this is very important slide on the different temperature requirements for the different vaccines. I will not spend much time on the details, but two important points from this um, slide. So it's a surprising fact, even for the experienced professionals, that freeze sensitive vaccines are most commonly damaged from the accidental freezing of the vaccine rather than the heat. So probably you will also shock that how come that vaccine are destroyed by freezing more than the heat itself. The second important fact, why it's happening, because in the freeze sensitive vaccine, due to improper storage, might be they have packed with the ice pack which are not properly preconditioned, might be the vaccine are touching the wall of the freezer. So whenever the vaccines, these vaccines stored below 0 degree centigrade. So in these freeze sensitive vaccines, which include DPT vaccine, hepatitis B vaccine. So what happens? We have aluminum adjuvanted in this now. So which actually participate while the temperature goes below 0 degrees Celsius. So the dangerous part is that you cannot differentiate between the freeze and the normal vaccines. Even by shaking, vigorous shaking, you cannot reconstitute. So this is a very important thing. So the last mile challenge. So the last mile delivery is actually the delivery from the regional health to the community level. This is where most of the supply chain failure occurs. Why? Because it combines of course, the temperature risk itself with the weakest infrastructure. I will not go into all these, the factors like the infrastructure, equipment, distance, human resource, finance, and data system, but I will focus on one thing, that these are not the abstract challenges. The important part of GSTP or important purpose of the GSTP is to ensure that every child is receiving medicines which is properly stored and distributed. So, and all, and the important message I want to communicate on this slide is that we need to design interventions that are based on the existing infrastructure. So what I mean to say, we need to provide solutions that work within the specified infrastructure, not the infrastructure which the countries, specifically the lower and middle income countries, they don't have. And let me explain in the next slide. So as I mentioned, so we need to provide solution that best suited for the available resources or infrastructure. And I'm pleased to share a few examples that it's not that innovation are happening in the vaccine distribution. For example, in Malawi, our African country, so where the normal kerosene refrigerators are replaced with the solar power vaccine refrigerators. So the pilot phase shows that there is a 3 times improvement in the vaccine quality. Then comes a very famous, trial. Not a trial even. It's now very much advancing in Africa. In Zambia, so the regulators has approved a controlled temperature chain that allows the meningitis A vaccine, Menafrivac, to be stored up to 40 degrees Celsius for the 4 days. And this enables vaccination in the remote area without having a cold chain infrastructure. So lot of reduction in maintaining the cold chain. So rather, they have approved this vaccine and the regulators are making sure that it's utilized within the 4 days. Next comes to the vaccine vial monitor. Very important innovation. It's a simple, low cost vaccine vial monitor are heat sensitive labels that shows whether the vaccine has been exposed to the excessive heat or not. Um, so far, I think since its innovation, it has saved more than 160,000 lives. Last but not the least, in Rwanda and Ghana, the blood product and vaccines are delivered through drones to facilities within 30 minutes. So the pilot phase has shown very promising results. It has reduced the product wastage up to 67%. Okay, so uh, this is, I think, the last slide for this session. So COVID actually stress tested everything we just discussed. I will not go into the much details, but I will share two important stories. So during the COVID, in one of the health center in California, they found that their one of refrigerator storing the Moderna COVID vaccine was out of order, and it was like Saturday late evening. So they don't have the alternate refrigerator. So what they did in the midnight, they have organized an emergency clinic. I think more than 830 patient vaccinated. This is smart. but this is not possible in most of the scenarios. Another example, during the initial rollout of the US Pfizer COVID vaccine, so due to labeling misinterpretation, one dose, there was a vial which comes with the um, 6 doses, but due to like not a clear like a labeling, one dose from a vial of six, I mean the dose of 6 doses was discarded. So let me explain again. So initial rollout, so of Pfizer COVID vaccine, the vial has to be constituted with the diluent, and but on the label it was mentioned that after the reconstitution you have the 5 doses, but actually you are getting the 6 doses. So how the health worker responded, they were discarding, or they thought that the 6 dose, after 5 dose, they have to discard this. For first 3 days, million of doses were discarded. And why this happens? A key lesson here, GSTP must be designed into the product and program design, not a retrofitted afterwards. No? Okay, we will skip this in the interest of the time. So next session is the E, which is the engineered products, which include cell and gene therapy supply chain. So vein to vein, very interesting phenomena. Uh, Let me share why the cell and gene therapy supply chain is different from the traditional pharma. In traditional pharma, the factory make millions of the tablets, many batches. From there, it goes to the warehouse, to the pharmacies, and then to the patient. Imagine if something happened with one batch or multiple batches. So usually that batch can be replaced by a newer batch. However, in the analogous therapy, for example, CAR T cell therapy, which is screening antigen receptor T cell therapy, you don't have such choice because the medicine is made from the patient own cell, and this is a batch of one, and it is for a specific person. Anything happen wrong, you cannot replace it. And let me quickly share why is that so. So in CAR T cell, the patient T cells are collected using a process we call leukapheresis. So to preserve the sample and prevent from the metabolism, the sample is stored at an ultra low temperature of minus 196 degrees Celsius. And we call this sample a cryopreserved sample. That cryopreserved sample then shipped to the manufacturers where the T cells are activated again, and they genetically modify those, test them, release them, and again the sample is converted into cryopreserved sample. And this process at manufacturer is 14 to 28 days long. So after again converted into cryopreserved sample, it's transferred to the or shipped to the um, facility, patient facility for infusion. So if anything happen during this supply chain, any wrong thing happen. So as I mentioned, we cannot redo it. And there are two main reasons. Because T cells availability in lymphoma patient is already a challenge. Even sometimes the quality of the T cells are not enough. So it very less likely that if somehow the first attempt was not successful, you were able to retake the T cell samples. It's like less than 1%. Also, the first time it cost more than 500,000 US dollar. So you can imagine it's already so much expensive. And if we haven't paid attention to the GSTP compliance, it will have a consequences not only on financial, but also might be lymphoma patient may not survive. Okay, so in the traditional pharma, there is a concept we call the chain of custody. Chain of custody means uh, that who will receive the product and when in the supply chain. So this is very much important, and this is important that chain of custody is not broken during the whole supply chain. And why this is important? Because you can imagine if unauthorized, stakeholders or personals deal your supply chain, there is a high chance it might be replaced with, some other products there could be. So chain of custody in the traditional pharma is very much important. But in cell and gene therapy, we have an additional requirement, chain of identity. So chain of identity means whose cells are there in that specific container. And this is very much important. Why? Because imagine if the error in the supply of custody in the traditional pharma, so might be the patient is getting different batch, problematic, but still manageable. But in cell and gene therapy, if the person or patient is receiving someone else genetically modified T cells, you can imagine it's a fatal. And how we can prevent this? There must be a unique identification on each and every step, not just unique identification, but there should be a real-time monitoring. And very importantly, we call a double check on every stage. There must be a double check. There is a no minor chance of error, that can afford the patient can afford. And also, last but not the least, usually we implement what we call the biometric verification at the infusion. Before actually we inject the infusion, there must be a biometric verification, making sure that this is for that particular patient. Okay, I will not spend time, but on this slide, but very quickly. So cryogenic supply chain. You remember from the previous slides, the cryopreserved sample. So they need to be stored at 150, minus 150 to minus 196 degrees Celsius. Of course, uh, uh, this facility is not available on every hospitals. Even the airline sometimes not accept this kind of a shipment because if there is a long transit time, we need a special dry shipper, right? As well as what we call the cryo charging station, because liquid nitrogen, it has a kind of a, you can say, a time frame or shelf life, and you have to replace or recharge your nitrogen for the long transit times. And 25% of the cost for cell and gene therapy is the logistic cost. Now, so as I mentioned, there are infrastructure barrier for this kind of a supply chain. And there's a 𝐃𝐚𝐭𝐞 supply chain study. So this is again in Australia before 2021. So for the CAR T therapy, the cells were extracted in Australia, shipped to the manufacturing facility at New Jersey, of course, manufactured and returned back to Australia. I think it's over 15,000 miles away. And of course, multiple international border crossing, custom clearance. Of course, there are cryo chain specific risk now. So but after 2021, so Australia built a local facility which solved the problem, but there was a huge investment. So think about it. Every time the investment is not possible, we have to strengthen our supply chain so the patient can receive the cell and gene therapy at the appropriate time. And very much important, connected with the previous slide, in the cell and gene therapy, time also matters. We cannot just say, okay, yes, we will do our best to deliver on time. But let me share two case studies, clinical trial rather, where due to the timing, the clinical trial fails. We call the Belinda clinical trial and Zuma clinical trial. So in the both are the for the CAR T therapies, T cell therapies. So for the Belinda clinical trials, the median vein to vein time was 52 days. Half of them, almost more than half, was just the transportation or cost. So what happened until the patient received this? The disease was already progressed and the trial failed. On the other side, Zuma 7 trial, the median vein to vein time was 27 days, which was 25 days less, and trial was successful. Another UK study found that 39% drop in the response rate if the time to infusion exceed 40 days, specifically for the lymphoma patients. So distribution delays has the clinical failure. This is not about the science failure, efficacy. This is about the speed of the distribution or supply chain. So not only uh, important we make sure the all the conditions during the transportation, but time is also important. Okay, so I think this is the second last session. It's linking the safety, distribution, quality with the patient safety. And this is very much important. I think this is the missing part in our whole system. So this means that linking your distribution quality with the pharmacovigilance, with the signal detection and risk benefit analysis. Let me share quickly. So imagine a biological product is perfectly manufactured in a facility and with all the specification or in good condition, it transported to a facility or hospital, but in between some unexpected thing happen, and due to temperature excursion, the product is partially, I want to emphasize the word on partially, and let me share why in the coming points. So the product, the biological product, due to temperature excursions, partially destroyed. Patient received the shipment. I mean, the hospital received, they inject to the patient. Overall, it looked good. The packaging is intact, but actually the product inside is not what it used to be. So what happens? The patient may experience reduced efficacy because, as I mentioned, it was partially damaged. So that's why still it's active, but it may initiate a partial response. And on the patient end, it's misattributed to a drug resistance or disease progression. We might be thinking, okay, might be this is due to drug resistance, but in fact, due to the distribution quality issues, it was partially degraded and was misattributed now with the drug resistance. So this physician in turn may increase the dose or may even switch the therapy. And usually in such scenario, the switch therapy is more costlier. As I mentioned that sometimes because the biological product is partially damaged, but damage sometime always that it's become inactivated. Sometime it's converted into another compound or another chemical, and that may initiate or trigger unexpected ADRs, which we never seen in the clinical trials. Probably you might heard about many reports that, okay, these are the clinical trials, which we never, the adverse drug reactions, which we never see in the clinical trials. And one reason is that due to the bad handling during the supply chain. So due to the partial conversions or inactivation or conversion into another chemicals, so the patient may experience unexpected ADRs. Increased immunogenicity, the patient may also experience this. Why? Because the new chemical may cross-react with the patient own proteins. And of course, the loss of sterility due to damage of the packaging. So the important thing here that distribution quality failure are invisible in the clinical system. Our PV system, pharmacovigilance system, may record as the adverse event, but we miss the root cause analysis. So the missing link distribution quality data is like rarely integrated in the pharmacovigilance system. And I will share like my finding. I have studied um, recently many GSDP guidelines, and in most of the guidelines, there is a zero reference about the pharmacovigilance. Okay, how we can integrate the distribution data with the pharmacovigilance data? So the pharmacovigilance data, the system are collecting adverse event report. On the other hand, the distribution system tracks the temperature and all the logistics, but they never talk to each other. These systems remain in silos. But there are many easy ways that we can connect. Like for example, there are 4 steps for the easy reference I'm mentioned on this slide. Number 1, batch traceability. Link the ADI report to the specific shipment condition. While you are evaluating like any AD reports, so try to link it with the shipment condition where possible. Second, wherever there is a alert about the temperature excursion, always pay enhanced PV monitoring for those particular batches. Number third, the geographical correlation. Cluster the adverse event reporting with the distribution route and try to find the pattern. Again, last but not the least, include your distribution risk and distribution pattern. And then let me explain in the next slide how we can do it. So this is the practical framework we use to include the distribution risk in risk management plan. So there are 5 steps. First one is the characterization. So product characterization, identify the critical quality attributes that can affect by the storage and distribution condition. For example, if you recall from the previous slide, so in the product characterization, you may find, okay, our vaccines are sensitive to the freezing temperature or freezing. So we must pay attention to those. Our product may be sensitive to the heat or humidity. So product characterization is very much important. Second, distribution risk analysis. So using the 𝐖𝐤𝐥9 methodology, identify the first map and then identify the specific risk. If we recall, the hands-off between the when one partner when the product goes from one partner to another partner in the supply chain, so identify those risks. Then importantly, monitoring strategy. Define the temperature monitoring strategy, how many times you will collect the data, and very importantly, you need to define the criteria. Temperature excursion threshold and minor deviation in the temperature. So there must be a criteria. Otherwise, for even a smaller deviation, you will get unnecessary alerts, which may jeopardize your overall efforts. Then comes to the signal correlation, the 4th point. So when safety signal emerge, always try to include the distribution data in the root cause analysis. And we have already seen how the distribution quality issues wrongly manifested as a PV signal. Last but not least, the continuous improvement. Feed the pharmacovigilance finding back into the distribution optimization. This is very much important. And in my experience, we somehow implementing all the 5 steps except the fourth one, the correlation. This is a missing link. Think about it. Okay, now we are moving to end of our presentation. Again, the key takeaway from this SHIELD framework, the standards we have studied so far. What is the GSTB standards? What were the 4 reference documents we talk about? GSTB is not an administrative checkbox. It's a patient safety imperative. Every supply chain must be controlled. Under the high-tech equipment, hopefully you like the concept of the stability budget and TOR or TOS, which is time out of refrigerator or time out of specified condition. Then we see under the vaccine supply chain, there are innovations already happening in the vaccine distribution. Vaccine vial monitors, drones, CTC. So then comes we studied the engineered products, the cell and gene therapy. We have seen how critical it is in the cell and gene therapy supply chain, not only control the overall the temperature, but also the timing is very much important. Then comes to the linking. We just studied that distribution quality failures are invisible to the clinical system. So we must try to integrate the distribution quality with the PV. Last but not the least, you must apply at least one thing from what you have learned today. Thank you so much for your time. And if you have any questions or you need any further discussion, you can reach out to me. Thank you so much.
Okay, that brings us to the end of the today's session. On behalf of IBPS DUHS, I'd like to thank Mr. Waqas Ahmed for sharing his expertise and thank all of you for joining us. For further queries, please reach out us to ibps@duhs.edu.pk. That's our email address. Thank you and have a great day, everyone. Allah Hafiz.